Hi, hello everyone. Thank you for joining us today for the first webinar of the GDRF. My name is Gilad Bennett. I am the father of Adam Bennett. Adam is an amazing 2.5 years old boy with the GRIN 2D mutation, the most rare GRIN among the GRIN family, as you're well aware. I'm also an active parent with the GDRF. I'm pleased to be here today to initiate our webinar sessions with the first webinar of today, hosting Dr. Tali GRIN. She will shortly provide an overview of the GRIN disorder genetic-wise and of the research angles moving forward. Before we begin, a few updates on the recent GDRF activity. We currently have several labs working on an active research of the GRIN gene and therapies for the mutations. Most of them are in Israel, but also worldwide. As you know, there are thousands of rare diseases, and we are very lucky to have gathered this unique group of researchers from Israel and worldwide that are willing to invest time and effort to find a cure for our children. This is very unique, and we are very lucky to have this. This group of researchers is led by the Vice Dean of the Medicine Faculty of Tel Aviv University, Professor Keren Avraham, and of course, Dr. Tali GRIN, who is here with us today. Also, we set the next event that will take place at Tel Aviv University on June 23rd. This is a GDRF conference that will gather all the researchers and physicians for a roundtable discussion here in Tel Aviv. You are all more than welcome to join, but we will also host it in a live webinar again on the 23rd of June next month. We also sent some invitation for worldwide researchers to join our discussion of the GDRF conference that will take place again in Tel Aviv University. The most important thing is that for all of us to join forces as soon as possible in order to get a research in place for our children and to find a cure in the upcoming future for our children. So please apply for membership for the GDRF Foundation and become a part of our group, because as you know, people are power, and as long as we will unite, we will find a cure as soon as possible. And together, I'm sure we will find a cure for all of our GRIN children. It is up to us, basically. So please do it just after our webinar today. And without further ado, I would like to introduce Dr. Tali GRIN to start our daily webinar. Please, Tali.
Hi, everyone. Thank you, Gilad, for everything you said. It's very exciting to be here after a lot of work that we've done to come up with this foundation, that it's cannot think of a better cause for this. I would like to start with this first webinar and to divide it for a brief scientific introduction of how GRIN disorder happen, what the research is all about. And the next half an hour will be answering questions from you, the participants. So I hope we'll have a fruitful discussion. So about the GRIN disorder, I know that we are not only from different nationalities, but we also know we have a different backgrounds, and some of us are parents that live with this GRIN disorder diagnosis for several years, and perhaps even attended several conferences. However, there are those that are new to this disorder and doesn't know much about what stands behind it. So I would like to say some words about the GRIN genes and the NMDA receptor. So what is the NMDA receptor? This is a very important protein. It's actually a channel that transfers information in the brain. It is very important for cognitive functions, for learning and memory. Actually, everything that we are doing is based on the activity of this receptor. From the time that we are born and we learn how to swallow, how to chew, then as we are toddlers, we are sitting and crawling, learning how to walk, how to talk, and then higher cognitive functions like mathematics, reading, problem solving, and on. We are learning and our brain is plastic throughout our lives. We are using this NMDA receptor. This is why this is a scope of many laboratories working on this NMDA receptor. However, not many are doing things that can come with solutions for problems with its activity. So the brain is all about communication. These are two nerve cells, and they are talking with each other. They are passing information in a special location called the synapse. Here in the synapse is the location of the NMDA receptor. So in this cartoon, you can see it's a channel that passes information between the cells, and it does so by passing electrical current. The receptor can work in the wrong way in two ways. First, there is the term gain of function that you probably heard before. It means that the receptor is overactive. It passes too much current. It is not tightly regulated as it should be. This causes a disease. On the other end, the receptor can be blocked, doesn't pass any information. So this is called loss of function. Both situations cause GRIN disorder. If it's too much activated and not controlled properly, or it is not passing any information, then sometimes you can get just the same phenotype, the same clinical symptoms as you all know, which means intellectual disability, developmental delays, hypotonia, epilepsy, autism, and more. So this, we can say it's a channel, it's a transmitter. This is a protein, and it's made up of 4 subunits. 2 of them are the product of a gene called GRIN1. And the 2 others are products of other gene, the GRIN2, that can be a GRIN2A, a GRIN2B, a GRIN2C, or a GRIN2D. Therefore, there are 5 genes that are involved in building this important receptor. Now, these are examples of combinations of this protein that is made up of these 4 subunits. You can see here 2 parts of GRIN1 gene and 2 parts of a GRIN2 gene. Now, I would like to make it very simple because some of you know genetics and others can't remember it from high school. Now, the gene is actually tell us how to build an instrument. Here we are talking about the protein, the NMDA receptor. It is essentially a coded instructions that knows how to combine building blocks, which are amino acids, to build the structure, the NMDA receptor. And sometimes you need several genes to make one receptor. This is the case in the NMDA receptor when we have 5 genes that are involved. Now, this code is made up of only 4 letters. It is G, C, T, and A. This is an example for a string of letters, while each 3 characters is coded for an amino acid, a building block. So if we look at the genome, we can divide it into chromosomes. There are 23 pairs, meaning 36 chromosomes. These are made up of 30,000 genes. And each gene is made up of these letters. So if we take it into account, the entire DNA is made up of 6 billion letters. What happens in GRIN disorder, there's a mismatch, there's a fault in only one of these characters. Only one of these 6 billions is wrong. So this tiny mistake is what made us here. Now, GRIN disorder can happen in several ways. Most of the cases are due to a change of only one letter, as I said. If we see here a normal gene, GRIN gene, we can see that the normal gene has a G. But in the second copy, which has the mutation, the G turned into T. This is called a G to T missense mutation. And missense mutation meaning we have now a different amino acid and the receptor doesn't work as it should have. These are most cases. There are also other cases of GRIN disorders that occurs when the same misspell cause no instruction. So there's no amino acid and the protein is shorter. It stops. This is a nonsense stop mutation. Now, there's also a possibility that instead of 2 copies, as we all have, we have only 1 because it was deleted. This is a deletion mutation. So you probably got results from of the genetic exam of your child, and you saw some sentences like words like likely pathogenic. What does it mean? It means that likely pathogenic, the genetic change, is significant and it causes a disease. Also, if it is written there heterozygous, it means, as I told you, we have 2 copies of each gene. Here, the problem is only with 1 copy, but this is enough to cause the disease. Also, the word de novo actually means new. What does it mean? The change occurred during pregnancy. It occurred in the child when it was developing at a very early stage. So all his cells all carry the same mutation. It is de novo because new, we as parents didn't have it. However, now that he has this mutation, he has a 50% to pass it on to his offsprings. So I told you that NMDA receptor is part of 4 parts. And in all cases of GRIN disorder, there's only 1 affected subunit. It doesn't matter if it's a GRIN 1, GRIN 2A, GRIN 2B, GRIN 2D. It's enough to have 1 mutation, and then ruins the entire apparatus. The entire receptor is now not working as it should have. So even only 1 GRIN subunit is faulty, still we have a GRIN disorder. This is called the dominant negative effect. This is also the reason why we should all come together, because there are 5 different genes, but the receptor is affected equally, and it causes a disease no matter which is the gene that is involved. Now, what do we have to offer for GRIN disorders? Basically, not much. This is the reason this GRIN Foundation was established. So if we're talking about gain of function, when the receptor is overactivated, then there are ways to block it, to inhibit it. Memantine is one drug known, but not if it was that simple, then we wouldn't be here. Memantine not only doesn't work all times, but also think that you need a receptor that is receptive and active. And if you block it, what can you, how can you learn things, new things? This is not something that we wish. Ketamine is the same as memantine, it's less specific. There's also magnesium. And as more than 50% of our children suffer from epilepsy, there's also use of anti-epileptic drugs, which are very non-specific They don't act on the GRIN genes, GRIN or the NMDA protein, and they have substantial side effects. If we look on loss of function, here we have nothing to offer. This is something that should challenge us. I would like to move on to the research, some background. So we know a bit what is a gene, what is the NMDA receptor, and how the different genes combine to create the NMDA receptor. So the GDRF was established to find a cure. And it was an association of parents, a non-profit association. When we want to give all the money to research, there are other good causes to organize us. It's either support, information, and more. However, for this, there are other Facebook groups. What we are focusing on is to find ways to change the future of our children and to make them fulfill their potential. There are many types of research. One of them is basic research. As I said before, NMDA is very important for brain development, brain plasticity. This is why many labs in the universe are working on this receptor. However, what they are doing is mainly understand how the NMDA receptor is working, where is it located, how it changes with time. This is very useful, very good information, but doesn't make the leap to help us as parents to find solutions for the problems that we are dealing with. Small part of basic research is functional analysis. In this part, we take the specific mutation and ask, how does it change the action of the NMDA receptor? Then we can have a result that will tell us if it's a gain of function or loss of function. But then we are also at a dead end. If we are a loss of function, nothing can be continued from here. If it's a gain of function, we can try memantine, but still, it won't be enough. So we are continuing to translational research. And this is what we are focusing in. In translational research, we would like to find ways to change and to help the disease, and to see if this can help human beings and not just animal models or cells in the laboratory. This is where we try to find initiations, to find new ways to tackle this problem. If we are lucky and we find a drug or a treatment, then we can continue to clinical research. It is a very important step where we'd like to see that the treatment is actually effective. We want to see also if it has side effects, how it works, how to give it. And this is usually done by pharma companies. It requires a lot of time, a lot of effort, a lot of money. If we want, for instance, a pharma company that wants to bring a new drug to the market, it takes about 10 years and $1 or $2 billion. So this is huge money. But we can help in the translational research. We can come up with, they call it proof of concept, something that was tested and was found to be good. And then we continue to the next step. So here in the GDRF, we will have, and we have an independent scientific board that decides how the money that we raise for research will be allocated, to which laboratory, to what area of research, and so on. We also want it to be full transparency. And as Gilad said, you are all welcome next month to see it with the brilliant scientists that are working on this project and the physicians that are taking it as their project and doing it so until now without any fee. I would like to mention two ways that we can tackle the GRIN disorder problem. One is the drug therapy and the other is gene therapy. What we are doing when we are looking for a drug, we want to make the receptor work as it should have. And we would like to change all the symptoms of the GRIN disorder, from intellectual disability to hypotonia and others. We have to consider the drug might have significant side effects, especially when we are talking about a disease that affects the brain, and also must be given for the rest of the child's life. When we're talking about gene therapy, it's a very new and fascinating area. Still, we have a lot to work to get into a gene therapy, but what we are doing actually is fixing the root from the cause. We know that GRIN disorder is a genetic disease. We even know exactly what is the gene and what is the specific letter that was wrong. Now we can target this treatment directly. And what's also fascinating about gene therapy, that it usually requires a single treatment. So it's a life-changing event. 6 years ago, gene therapy for GRIN disorder was non-existent. But there is a revolution that made us come up with new tools that can manipulate the genetics. Not only can we identify diseases, you know, a few years ago, nobody knew that GRIN disorders, what causes it. Now, not only that we know it, we can also change it. So I'm going back to drug therapy. What our strategy is to create an NMDA receptor, an artificial one, in the lab. It will be made of GRIN1 and GRIN2, obviously. And then we use it in a robustic way to screen for thousands of FDA drugs. What will be the result of a successful screen is a few drugs that we can directly give it to children because they are already on the market and we know exactly what are their side effects and what to expect. The other pathway is the genetic therapy. And here, as I said, due to evolution that occurred in the last years, we have now the tools to change the exact cause of the disease. How can we do it? This is a scheme of CRISPR-Cas9. This is an enzyme that can, with a guide, can reach the specific location in the DNA to find the mutation, the specific location where the change occurred in the child. Then it can cut the wrong letter and put the right one instead. We have to do it where the problem is, meaning in the nerve cells in the brain. It's not something easy, but it is doable. We are using AAVs. This is adeno-associated viruses. It's not a real virus. It's a vehicle. It can deliver the genetic tools where we want, inside the cells, inside the neurons. And then we can change their genetic background. Now, AAVs are proved by over 100 clinical trials to be safe. They are not causing a disease, and they just a form of transportation to get us into the right location in the cell. A very important step is to see if the treatments, either drug or gene therapy, are effective and safe. And this can be done in 3 ways. Sorry. Let's go back. One is using cells in the laboratory. The other one is using animal models. And of course, eventually, human beings. Another very fascinating technology is the ability of current researchers to take cells from a person, make them into all types of cells that want to be tested, and to try drug or different treatments directly on them. Here is an example how we take skin cells from a child with GRIN disorder. We produce stem cells. We convert them into the nerve cells these are the cells that interest us. And not only that, we can make a little brains in a dish. It calls brain organoids. They live for more than 3 months, and a very good model to test diseases of the nervous system. Animal models are also a very important tool in research. And until recently, we had no such a tool that carries human mutations. And there's a lot of progress now in only the last year, when we are having a GRIN1 mouse, the first one that has human mutation. We also, here in Israel, made a GRIN2D mouse. We are now at the process of studying that it is the right genetic manipulation that we want. The same is true with GRIN2B. In a few weeks, we will know if we have a mouse suitable for our experiments. And in the near future, we plan to create also a GRIN2A mouse. I should mention that in the past, it took around 4 or 5 years to create such a model. Now we can do it in only a few months. It is a real revolution. So after the background, the scientific background, and what we want to do, I would like to tell you where we stand now. You must know that science takes a lot of time, a lot of energy, a lot of resources. What we have accomplished was done with the help of several laboratories in Israel and abroad in a very short time, only several months since everything started. So as Gilad mentioned, we are fortunate to collaborate with brilliant scientists that they have their heart in the right place and really care about our children. They are located at 6 different laboratories. Each laboratory has its own specialty. I should mention one that is expert on NMDA receptors and human mutation. They can do functional analysis. They can create the assay for the drug GRIN. Another laboratory is expert on making nerve cells from skin cells and brain organoids. Another laboratory is focused on gene editing and gene therapy of several neurological diseases, now GRIN as well. They know how to use AAVs, and they have the ability to make genetic changes in GRINs and in other genes in the brain. So this is only in a nutshell. Now, they are from different universities, and the vice dean of the medicine faculty in Tel Aviv took this project on herself. She's a great woman to support us, but we also need physicians, and we are working with neurologists from Sheba Medical Center. It's one of the 10 best hospitals in the world, mainly because there are good physicians there, and they're also good researchers, and they're using new methods to treat human diseases. So they are with us, and they all know about the GRIN children, that, as you know, not many physicians, not many neurologists know about it. You are, as a parent, even know much more than them. You have to educate them. Not only in Israel, but also in other countries, we are forming connections and collaborations in Canada, France, Poland, and more, because we understand this is a very huge project, but it has such an important cause, and we do believe that the future is here. So where do we stand? We are actually in the middle of building an assay for drug GRIN. We have the entire machinery to test drugs, to test FDA drugs. We have the entire robotic equipment. We already did half of the assay. As I said, we need to express both the GRIN1 protein and the GRIN2. So we are successful in the first part. So we have the GRIN1, we have to combine it with its partner, and then we have the full receptor, and we can start the screening. The screening itself will take only a few weeks. And then the next step is, even though it's an FDA drug, I would like to see how it works in an animal model. And for that, we have the GRIN mice. So we have several GRIN mice. some of them already proved to be with the right mutation. We need to make them appropriate, to make enough of them so we can test them, to do a behavioral test, motoric test, and to see that first they have the disease, and then we can make a change. And then, meaning testing the drugs on them. For the second and exciting part of personalized medicine, where we take a specific child and his specific mutation and do all these assays in the laboratory in a dish. So we have several children that we already have the cells stored. We have a bank of these cells, and we're in the process of making them into nerve cells. For that, we need Helsinki. We have to be in a hospital for doing this, and this is after we will finish with the bureaucracy steps. Then we're ready. We have an excellent laboratory that is specializing in brain organoids, and this will we will test therapies directly on the cells. And for the third part, the gene therapy, this is what actually will make the change, will make the real treatment in these disorders. So we already have the entire tools to do the change of one letter for the other. We have the enzymes, we have the viruses, we are ready, we know how to design this treatment. I should mention that even though every child has a different mutation, the main tool is the same, meaning the enzymes are the same, the viruses are the same. There's only a very small difference that distinguishes one treatment of a child from the other. And when we are ready to use these genetic tools, we will test it both on the mice and on the human cells. So for good science and for research, we need good people, we need scientists, we need physicians, and we also need to pay for what they are working with. So until now, the research was in the academy based on grants, and what other resources are government investments in science, also initiations of biotech companies, but we also have to have some help from other organizations in order to help our children, and part of it is also us. We are those that can change our future of our children. So this was the first part of my talk. I think that we are ready for the questions.
Q&A session. Just before we begin, I want to relate to some of the things that Tali has mentioned. So as you saw, there is an active research going on at the moment in several universities and several researchers. In order to continue with the research and not to rely just on the goodwill of the researchers, we need to get the funding in order to speed up the process, because our children, unfortunately, doesn't have the entire time in their hands. So we need to speed up the process. So in order to do it, we need to motivate the researchers using funds that we can gather from personal, from our families, from different organizations, and from other locations.
אז תעשה אונליין.
So this is highly important in order to push forward the research. You can find further information on the current research, other activities on the GDRF website. And we will shortly open the donation section in the website. For that, we have teamed up with Kav L'Chaim and JGive. They are social associations with more than 40 years of experience on raising funds for complex disabilities and rare syndromes. And they will help us to raise the funds needed for the project. You can check their website online, and we'll share it on the website. Without further ado, let's go on to the Q&A session. Back to Tali, and we will continue the discussion.
Okay, so everyone can ask a question. In the bottom of the screen, there's an icon, and you can press and ask a question. I can start with questions that were transferred by mail. So the first one is from Christian. She asked, does CRISPR work as a treatment method for GRIN1 mutations? So, as I mentioned in the first part of the talk, we have the tools to use CRISPR and change the GRIN1 mutation. Obviously, it depends on the type of mutation. We have several options. For instance, if this is let's say, a G to A mutation, there are definitely tools to make it back from A to G. We have to know exactly where the location of the mutation. So we need the specific information about the mutation. Then we design the treatment that, as I mentioned, it's not that different. Most of the part of the treatment are identical between the children, but there is a very minor and important change, and for that we need to know exactly this type of mutation. So definitely CRISPR can work. There's also other ways that CRISPR can work in different mutations, for instance, for a deletion. For a deletion, it is possible to increase the amount of the gene product in a different way. So again, we have to see if the mutation is a missense, a nonsense, a deletion, to know exactly where it is on the sequence, on the gene. And then we can design the treatment, and then we will test it as one of the methods that I already talked about. So there's another question of Micha. He's telling us about his 5-year-old daughter that just recently found out that she has GRIN2B. So they would like to talk about further tests that should take and the connection with the focal point in Switzerland. Okay, so I think that the GRIN2B Facebook group is a good place to start about connections, then because it combines us people together. We don't have a list of the doctors or those that treat GRIN2B in every country. This is information that it's important to get from the Facebook groups. I think that the first step is to know what type of mutation it is, and to decide if you would like to join and to be one of those that support science and to work about this specific mutation. Another question from Bianca, and she asked, how far away are we in terms of years from a cure for GRIN1? Are there medications or solutions to help us with the intellectual disability aspect of the mutation? So, well, I'm not a prophet, But I can say this. 10 years ago, no one could have talked about a cure for GRIN 1. In 10 years from now, it will be common practice. This will be, I will give you an example. This year, not recently, our government decided to support an initiation of gene editing. Gene editing is something that a few years ago was a dream. Now, the government is willing to pay several $million for companies and for combinations of academic researchers and drug companies together to pursue this goal. So if government puts money on something that once was a dream and now is becoming reality, I think there's a good chance that in the very next few years, we will have tremendous results from genetic therapies, genetic editing. The problem that is we are GRIN disorders. There are 7,000 rare diseases, most of them are genetics. So we are only one of many. And if we don't push gene editing, gene therapy for GRIN, then the resources will go for other genetic diseases. So we are few, but we are determined, and I think we should be very optimistic about the future. So another question from Jan. I will make it short. Are the drug trials mostly for those GRIN to be individuals who have seizures? Our little one is 5 and has no other major medical issues. She walks but doesn't sleep, says no words, and does nothing proposal at all. It seems no incoming or outgoing messages are being processed at all. Do most of the GRIN to be children have multiple medical issues? So I think that every GRIN to be is different, and sometimes a GRIN to be can be more like a GRIN 1, and as I mentioned, we are all making the same receptor, so the difference between the specific genes is less important. What is more important that we all have a problem with the NMDA receptor. So the drug trials are not only for seizures, definitely not. There's a large part of children that don't have epilepsy and have intellectual disability. I think the major problem with GRIN disorder is the intellectual disability, because epilepsy, yeah, of course we want to take control of it, but with the intellectual disability, it means that the child will be always subjective to others. He won't be independent, he won't fulfill his potential and be independent in his life. I think the issue here is to tackle especially the intellectual disability. Now that we know exactly the genetic cause, we can say that if we change the mutation to a normal one, there are good chances that it will work on every aspect of the GRIN disorder, not only epilepsy, but also intellectual disability. Right now, we have no solutions for intellectual disability. This is the major goal of the GDRF. Okay, I'm moving on to a question of Stacy. She asked, how can parents with bioethical concerns, religious belief, and others might handle participation and application of current and future research? So this is a very big question, and I don't think this is the scope of this webinar. I think that people who don't want to use animals as tools for promoting science and medicine, they have a right to think so, to live according to their beliefs. Me personally, I would like to see that a specific drug was tested before I give it to my child. Also, I should mention that if we look at the personalized medicine, this part, when we use the human cells and work in a laboratory and test treatments directly on it, I think there are less issues of religious or ethical problems. So this path, I think, can be suitable for people who don't want to participate in some aspects of research. Okay, another question from Marita. She asked, regarding this personalized medicine, the drug that will come from the result of the screening, will it be a trial on the patients for these drugs? Are you collaborating with hospitals, doctors that have these patients with this different mutation in the different countries? Kind regards. So, if we're talking about personalized medicine and we're talking about drugs, so if a drug is an FDA drug, there's no need to do a clinical trial because this is supposedly a safe drug and can be given. About collaboration with other hospitals and doctors, so first we need to find something that works and then we can offer it to other hospitals. We would like to have free information and free exchange of data between other hospitals. I think in the Atlanta conference, it will be important to speak with others and to make a larger network for that purpose. So I really hope that we can have good results with the screen and then we can continue on. Okay, another question from Karen. She asked, which studies seem most promising and financially supported, gene editing or drug intervention? Okay, so the second part, financially supported. So until now, there was no support whatsoever, either for the drug screen or the gene editing. Now, what seemed most promising? As you can see, we're working in parallel ways, both on drug and on gene therapy. There are pros and cons for each road. I don't want to say my personal belief, but what comes first? If the drug is first, then it will be used. If gene editing is first, then this is where we are going. But we are trying to see it in a broad perspective. So sometimes solutions come from unexpected locations. Another question from Janet. Okay, she writes, Adam is 27. If a drug remedy such as L-serine worked, would he need to learn everything from scratch? Would he need therapies? I'm pretty sure he would. What about speech? What are the chances of sounds developing? It all sounds so unreachable at that moment. Okay, so again, this is a tough one. We cannot speculate what will happen in the future, but I can assure you that we are constantly using NMDA receptors. We are learning new abilities. We are learning new ways to navigate in space, in time. So we need a receptor working when we are 1 or 27 or 40 years old. This is the reason that we believe that if we change an active receptor that is mutated and make it normal again, then we can restore abilities. If you see the children that have problems with the NMDA receptor, but still has some activity of it, still learn, although very delayed, they learn to do new things, their motor coordination improves, they learn few more words, then it means that what's left of the receptor is working. So imagine what happens and if we can restore full activity of the receptor. Obviously, it will have to learn things again, but I wish it would happen, really hoping so. Okay, another question from Natalie. Is there any point for a teenager to start any sort of medication you will find, or is it too late? Again, no, it's not too late, definitely not. Okay. Okay, question from Bridget. My daughter has a stop codon, and at the lab at Emory was not able to determine gain or loss. From the test they ran, they all came out in normal range of functioning. But my daughter has epilepsy, intellectual disability, mild hypotonia, developmental delays. Are there other tests to determine gain of function or loss of function? Or there another issue that is not defined by gain of function or loss of function? Okay, if she has a stop codon, it means she has less amount of the receptor, because the protein, we have one normal copy, and the other copy is causing a receptor to be shorter and non-effective. So this is why she has these symptoms. What we can do, actually, it is a loss of function. What we can do is to make the one normal copy of the gene express more of the NMDA receptor to increase its production. And this can be done mainly in gene editing techniques. So I don't think there's any point of making more tests. I think there is a point to start finding a specific treatment for this type of stop codon. Okay, another question from Bridget. What are potential risks of gene therapy? Okay, so gene therapy, there are potential risks, but I think maybe we should discuss it in another seminar, because I have to explain several aspects of gene therapy. There's also, it is a very general description, gene therapy. We are trying to move in several directions. So for each and every one, there are advantages and disadvantages. So I think we should discuss it hopefully in another webinar. A question from Dave. Are you taking tissue or blood samples? Okay, so this is a good question. We can make nerve cells both from blood or from skin. The results are much better from skin cells. This is a much better material to work with. Now, we have to pass, as I said, the Helsinki rules, and when this issue will be settled, then we are starting, we are also already have cells waiting for this. We will start producing those specific personalized brain cells. So it will be, we will follow up on this matter. Maybe it will be easier to take these samples from here in Israel, because there's a lot of rules and regulations and restrictions of sending material, human material between countries. Okay, another question. So it might need to be, people have to be physically attending Israel for making these samples. The procedure is very simple, it's very easy, it doesn't cause any damage or pain. I'm as the dermatologist doing it every day with no problem. The problem is only the bureaucracy about sending something from different country. Okay, for another question. How is it possible for a child with loss of mutation to have epilepsy, what the cause of this, because as I understand, this is normal for children with gain of mutation. Let me see if I understand it. Yes, loss of function also have an epilepsy. It's not something that occurs only for gain of function. This is a well-known fact. It's very intriguing. It's very, it's a bit confusing, but this is a fact. Loss of function also causes epilepsy. This is because when a receptor doesn't work, there is imbalance in the brain, imbalance between activation and suppression. And when there's no normal suppression of electrical activity, then epilepsy occurs. Another question from Natalie. Oh, I answered this already. Dave asking, are there indications for CRISPR for GRIN to be efficacy? CRISPR is definitely something that we are using as a major tool in gene therapy here in this foundation. Another question from Yolanda. How will I know my child is gain of function or loss of function? And is the new method of changing the code of the gene for both gain of function and loss of function? So for gene editing, it really doesn't matter if it is a gain of function or loss of function. It's really, it works for both situations. Now, if we want to know if the child has gain of function or loss of function, one way is to go to a laboratory like in Emory and ask for a functional analysis. It might be, they are giving grants from the NIH for this. So there are also another laboratories in Europe that are doing this, this service. Again, if we know that, if you know that the child has gain of function, it is possible to ask for memantine. And if it's a loss of function, it won't be effective. But this is the only thing that gain of function or loss of function will tell us. Still, I think that we need to correct the problem from its root. Okay, another question from Sam.
And that's one.
The last question. No, it's not the last.
No, that's because we are.
Okay, we don't have enough time, so we have another question.
No.
Just a minute. Okay, I already answered it. Okay, so it was already answered before. We are going back to Gilad.
Okay, thank you, Tali. Thank you, everyone, for joining us for this first webinar. We will hope to do it again in a few weeks on other subjects that will be raised during the recent time. You can search our website, greengene.com, for more information on the researchers, and other interesting topics regarding our children. And as I mentioned before, we will start the funding soon, and please stay tuned for updates in order to see how you can donate. Now you can gather everyone you know in order to donate to our cause. And of course, join our membership as part of the GDRF Foundation. This is very important that we are united. Thank you all. Have a good night or good morning. And take care, and we will see you soon on the next webinar.